Thank you very much for the kind introduction. It's a pleasure to be here. So we're going to spend, I think, the remaining of the afternoon together. So I'm just to start with, this is where I'm based. This is where we are, somewhere here. And this is more in detail where I'm based. So I'm based at Utrecht University. Utrecht is pretty much in the center of the Netherlands. This is our academy building. The university is officially dates from 1636 when it got the university title, but there was a school before that which dates from the 12th century something, so it has an old history. And this is the building where I'm in. This is actually an NMR center, nuclear magnetic resonance, and you see some of the machines that are here. So I'm doing computational work in the middle of experimental people. This is actually my office up there. So we have a strong infrastructure for NMR. We are a European facility. We have our own small, modest server. This is where part of the Haddock computations are running. And we have access to distributed resources across mainly Europe, but also the world. And we have access on paper to about 100,000 CPU cores. This is still a small number compared to what's standing next door here. But this is very useful for providing services to the community. So today we're going to speak about molecular machines of lives and how to model those. So we have our genome information, the DNA, which encodes for information basically, but that's the DNA in itself is kind of boring. It's the storage of life. Some people get very upset if I say that, but if you think of computers, DNA is the hard drive. Proteins are the CPUs. Proteins are doing the work. Of course, you need both memory and you need both CPUs to be able to do anything useful with a computer. The same is true in life. And proteins are not working alone, but they work by interacting with other biomolecules or small chemicals. And this is the domain. So we go from genome to proteome to the interactome, all the interactions that are taking place in a cell. So basically, what you want to understand or what we want to understand and model is the social network of proteins, because they regulate a lot of life or what's happening at a cellular level and communication between cells as well. And what you see here is the center of Utrecht. So Utrecht has that's the old Rhine, a canal going through the city. Maybe some of you have been to Amsterdam, and Amsterdam also has water, but Utrecht is much better. And there's a good reason why Utrecht is much better than Amsterdam, is that if you look at this picture, there are terraces on the water level. If you go to Amsterdam, there's the street, and then there's the water, and there's no terraces. So in Utrecht, you can enjoy a nice meal at the water level. And this is where you see a lot of macroscopic interactions taking place between people. And in the cellular life, you will have microscopic interactions between biomolecules. So how can we study those interactions? That brings us to the structural biology of interactions. So on the right side, you see some of the experimental methods that allow you to get information about molecular complexes. And there are three methods that can give you experimental information at atomistic level. X-ray crystallography is the oldest of all, NMR, and cryo-electron microscopy. And these are the only methods really that can give you atomistic coordinates of your systems, experimental coordinates of your systems. But there are many other methods that are giving you pieces of a puzzle. Small angle scattering, SACS or SANS if you do neutron scattering or X-ray scattering. Mass spectrometry is giving you also pieces of a puzzle. You might measure a few distances using crosslink MS, or you might measure regions that are affected by binding. So you might do a simple biochemical experiment, do mutations, run a gel to see if a complex is formed or not. All this provides you pieces of information. And you need then to combine those pieces of information with computational methods in those cases to build 3D models of your complexes, which brings us on the left side to the in silico world. And there, when you look at that interactions, there are a number of methods over the years. These are the classical methods. So homology modeling has been there for a long time. So there you transfer information from non-protein structure in a PDB to the complex that you are looking at, provided there is a homologous complex. In that case, you might want to do molecular dynamics. We've heard about that in the morning. Although simulating association of proteins is a rather slow process. And what we're going to focus more when we speak of integrative modeling will be docking. Of course, things have changed in the recent years because of the outcome of AlphaFold. Very quickly, AlphaFold was designed to predict protein structures, not per se complexes, but very early on, people realized that you can also predict complexes. The first version of AlphaFold 2 release, people were playing tricks, adding a flexible linker in your sequence and predicting a complex as if it was a very big protein with a linker. And then DeepMind released AlphaFold Multimer. And after that, you've seen many different versions. AlphaFold 3 is now out. There's OpenFold 3, there's Chai, there's Bolts. There's a lot of techniques that allow you now to study complexes also from an AI perspective. So before moving more into integrative modeling and Haddock, I want to spend some time in discussing what AI has been bringing to the field of interaction and structure prediction, and also show you what are the limitations that are still there. Not everything is solved. Who has never used AlphaFold in this room? Okay, so that's a few people, and you have so that's not a lot. It means most of you might have used AlphaFold or something. There's also a big database of structures, so you don't even need to do prediction these days. There are more than 200 millions of structures that have been modeled using AlphaFold in the AlphaFold database. So that's a starting point. There are no complexes there. So if you are studying complexes, you will have to model them if you want to use AlphaFold. So we'll start with the critical assessment, CASP and CAPRI, and describe the rise of AI and AlphaFold in that context. This is based on slides that I got from Mark Lansing. Mark Lansing is an assessor in CAPRI. I'm currently the person running, the director of CAPRI, and SG has been an assessor in CASP. So critical assessments, so there are two main experiments. There is CASP, which stands for Critical Assessment of Structure Prediction, and there is CAPRI, which stands for Critical Assessment of Predicted Interactions. So CAPRI focuses on interaction, and CASP historically started with structure prediction of single proteins. And since several rounds of CASP, now the focus is shifting more toward complexes. So CASP was born in '94. It runs basically every 2 years. And there is a prediction season of 2 to 3 months where groups are spending a lot of all their efforts basically in those 2-3 months to predict structures. And there's a round of CASP that's going to take place this year starting sometime in May. CAPRI is a bit younger, 2001. And the predictions here take place on a rolling basis. Basically, whenever we get targets from experimental people, we organize a round for CAPRI. And we team up with CASP in a CASP-CAPRI round. So if you're interested in testing your methods blindly, there's a round of CAPRI which is going to start in a couple of weeks from now. You can register now. We have 8 targets in that round which are antibody-antigen complexes. So you can put your skill to the test. And since 2014, basically, we are teaming up, CAPRI and CASP are teaming up to do the assessment together when there's a CASP season. Just to show you that assessment is a big deal, these are some statistics from CASP 14. So there are about 200 prediction methods that are put to the test. So this, since the early days, was the way to really blindly test the software and your methods. Because you don't know the answer. When you are developing methods, you have to benchmark the methods, to tweak the method to get the best results. But somewhere in the back of your head, you might know the answer and you might take choices that brings you to the answer. In a blind experiment, you cannot take those cutoffs, so those shortcuts. So this is the real true assessment of the performance of a method. So it's a lot of, 200 methods are being tested, 350 predictors taking place. It generates a lot of model, lots of data, and a lot of, every year or every second year when CASP is running, there are a number of people who are doing the assessment, and this is changing. While in CAPRI, it's always the same people doing the assessment. So it's a big thing. So now we go to 2018, the dawn of AI, basically. So this is what you see here is some plot coming out of the CASP analysis, where on the x-axis you see predictor groups, and on the y-axis this will be some measure of the performance of those groups. And the higher the bar, the better you are. Okay. And this was 2018, and there was one bar which was slightly better than all the others, but at that time that was a big jump, okay? And this was AlphaFold 1. So 2018, AlphaFold 1 was released. The code was kind of made public, but you could only repeat the predictions from CASP. So it was a useless piece of code at the time. But this was already something, and this was a new predictor. This was Google DeepMind entering the field of protein structure prediction, and nobody knew that they were doing anything there. 2 years later, CASP 14 now. What you see here is the same plot. The two bars here are the Baker and David Baker groups. So David Baker has been participating since the early days of CASP, and his software, with his software Rosetta. Some of you might know about Rosetta, and his groups have always been among the best one, and it might shift from time to time. But it was not the full picture, because what happened in 2020 is this. And so this was AlphaFold 2, which was way better than anyone in the field. So everyone was blown away. The prediction performance, so one thing about CASP is that, so they always made these plots all the years. So if you go back to the old years of CASP, you will always see this kind of plots. So there was always a winner, but you could be the winner of the crap, meaning you are doing slightly better than the others, but your prediction are still crap. Okay, so there's always a winner in CASP. But this time, this is not only much better than everyone else, but the targets were right on spot. Okay, so the prediction were very accurate. So people say, wow, the structure prediction problem is kind of solved. Of course, DeepMind, Google, with all their PR machinery, made a lot of noise about it. So it was all over the news, method of the year in Science and Nature. Boris Johnson, at the time Prime Minister of the UK, tweeting about it. So this was a big thing. At the time also people were saying, well, this has solved the folding problem. Folding is a different thing than structure prediction. Okay, when you speak of folding, it's more the process that the protein sequence coming out of the ribosome takes to fold into its final three-dimensional product, a 3D structure. What AlphaFold is doing is not predicting how a protein folds, it predicts the end point of the folding. Okay, so it's a structure prediction, which it's doing, but not the way that the protein folds. There are many diseases that are related with misfolding events or things happening during the course of folding, and you cannot study those really well with AlphaFold. So you will need methods like molecular dynamics, for example, to study, if you want to study folding processes. So this was we were 2020, the breakthrough, and then in 2024, they got the Nobel Prize for this in chemistry. So this only 4 years after the work was done, the Nobel Prize was attributed to Google DeepMind, Demis Hassabis, and then John Jumper. So Demis Hassabis has I think is a PhD in neuroscience. John Jumper is a chemist by training, quantum chemist, and he is the one who brought basically the molecular aspects into DeepMind and helped in the development of AlphaFold 2. So they got it for structure prediction, and David Baker was also part of the Nobel Prize at the time, and he got it for protein design, although he's been in the field of structure prediction since the early days, but they had to distinguish between the two. Anyway, this was a big change. In CASP 14, Capri was also participating, so we had CASP Capri, and at the time there were 14 complexes. So this was not only about predicting the structure of the individual proteins, but there were also complexes, okay? And what you see here now are the people participating in Capri as part of CASP. So these are different groups there. We assigned a score, so this is now Capri metrics, and in Capri we distinguish between high quality predictions, so this will be like a crystal structure basically. You are within 1 angstrom RMSD from the right answer. You will have a medium quality prediction, and then you have acceptable. And depending on the number of targets or complexes that are correctly predicted, we assign a score, which also depends on the quality. The triangles tells you how many complexes were correctly predicted, and the bars show you the score, which is the combination of high, medium, and acceptable. So the best predictor, Seok from South Korea, got 9 targets. And then you have a number of predictors that are at for the 8 targets correctly predicted, okay? And then it slowly goes down. Now, at the time, AlphaFold was not used, so AlphaFold appeared in Cas14, so they didn't apply it to complexes at the time, but we took basically AlphaFold to multimer and then predicted the targets of Cas with AlphaFold 2 multimer. And this is what AlphaFold 2 would have done using it off the shelf. So if the number of targets that it's correctly predicting is not different than what the human groups have been doing. Okay, it's predicting 9 targets out of 14 correct, and the other one are wrong simply. But you can see from the bars that the quality of the prediction is much better than what the human groups have been doing. Okay, so AlphaFold can also deal with complexes, although it has not solved everything when it comes to complexes. So there's still work to go. So then came Cas15. So this is 2022. This is where AlphaFold stands in Cas15. Just, again, AlphaFold multimer taken out of the shelf. Of course, AlphaFold was there, and pretty much all the groups now were using AlphaFold to do their predictions. So everyone on this region is using AlphaFold or some version of AlphaFold or some slight modifications. So you can see that now groups start doing better than the baseline of AlphaFold. And there are 3 groups that are jumping out compared to the others. So Walner's group from Sweden, Pesi Folding, and Venklovas from Lithuania. And they did slightly different things to improve on top of AlphaFold. So Walner did a massive sampling. You know, if you use AlphaFold multimer, the default number of models that it will predict will be 25. I think it was 5 at the beginning, and people increased a little bit, so you will get 25 models. And what Walner was doing in Sweden, they had just installed a big GPU clusters, which was not yet open to the public, but Walner has access to it. So he could run, instead of running 25 models, he was running 6,000 models with different. So it was a massive. So you just keep repeating AlphaFold predictions with different seeds, and this improved basically the success rate. Okay, so this was major sampling. Of course, it comes at a major computational cost, so you need a lot of computing to be able to do that. The other groups that improved things was doing standard number of predictions, but they start playing with the multiple sequence alignment. So AlphaFold is using multiple sequence alignment to extract coevolution information that gives you kind of distance information within your proteins, but also between your proteins. And you can do smart things in generating your multiple sequence alignment. And by doing smart things, you basically make the signals appear better out of the coevolution signals. So this was for pruning, and then Venklovas, he has a long history of participating in Capri, and he's very good at scoring. Scoring is basically you have a set of models, and then you have to fish out which one is the best. And they have their own methods. So Venklovas had very good scoring on top of AlphaFold predictions. So basically, if you look at the success rate, so this is again some kind of Cas measure, but if you look at the success rate, if you run naive AlphaFold 2, and so here you will get in the order of 30% success rate, and the top performers could increase their success rate to almost 90% when you look at the number of targets. So then it's still AlphaFold, but you can do things better than just running it naively. Does it mean that everything is solved? No. So these are now some examples from Cas15 and Cas16. So in Cas15, so we are now at 14, 15 is 22, and 16 is 24. Okay, so here you see some of the things that did not work very well. You find nanobodies, antibodies. Multiple sequence alignment are important to extract coevolution signals. It means, and coevolution signals comes from when residues are correlated or are mutating at some point in evolution in a correlated manner. And it means that there must be some conservation. When you speak of antibodies or nanobodies, by definition, those biomolecules are evolving extremely fast in their binding regions because they have to adapt to immune threats, okay? So there is no conservation, and it means that the signal for the binding regions of those molecules is not really there. And that caused trouble for the predictions. There were other things that were, domain swap, that's a tough one to predict. Multiple conformations, that's a hard one. And in Cas16, there were quite a number of RNA targets as well, protein RNA complexes, and pretty much there, none of the prediction. So AlphaFold 3 was out. AlphaFold 3 can deal with RNA and DNA. AlphaFold 2 cannot, it's only proteins. None of the prediction of AlphaFold 3 made sense when it comes to RNA. Okay, so the best predictors were still human groups with a lot of expertise and experience in modeling RNA. And we can still do much better. Okay, so it's not like the problem is solved, so there's still space there. So this is Cas16. Okay, so in Cas16, again, looking at interactions, not single proteins. So we had 34 complexes, which formed 47 interfaces. Okay, so this is, again, the ranking for the complex prediction in Cas16. So this is what you get from the submission. So the best predictor is Kozakov. He's someone who's been doing docking for a long time, but he's now using also AlphaFold for this. But there are some issues. So if you look at some of the predictions out there, what was also happening here is that people were now running these massive predictions with AlphaFold. So there's a method called massive fold, which runs AlphaFold with different settings and generates 6,000 models per complex. And then you have to fish out which is the best model out of the 6,000. So if you take the naive AlphaFold score, and for complexes, it will be not only PLDDT, but IPTM. The oranges, the crosses here, are basically telling you which model did AlphaFold select as being the best. The line shows you the quality of the model, order based on quality, from non-acceptable, yellow, to blue is medium, is acceptable quality according to Capri. Green will be medium, and then there will be a red point there that you can barely see, which will be high quality prediction. So you see, in this particular case, it's an antibody peptide complex. AlphaFold picks a model which is of medium, which is acceptable quality, so it's okay, but not very good. And in the data set, there was a much better model. And this is even worse. In this case, the best scoring model according to AlphaFold is a wrong model, but in the data sets, they were high quality models. And we were not able, or the community was not able to pick those out, okay? So if you look at that, let's take all the models from massive fold, and if we had the perfect scoring function, if we were always able to select the best model, this is the performance that we could get. If you combine on top of that all the predictions from all the predictors, so we take everything that the community has predicted, and we have the perfect scoring method, we can still improve things a lot. Okay, so this is what we are achieving now, and AlphaFold is here, that's the CollabFold baseline. This is what the best predictor do, and this is what is possible. So there is still work to be done to reach that level. And it's not a sampling problem, this is more a scoring problem. How do you identify good prediction out of your set of prediction? Okay, so if you are in computational structural biology, don't be too afraid of AI and AlphaFold. They have not solved yet all problems. So we still need probably some good old physics to deal with that. This is true, so we can improve things for many different classes. So it's not only antibodies that have problems, but you see here heterocomplex, these are homomeric complexes, these are large complex. The antibody ones are here, and nucleic acid complexes are here. So everywhere you can see that the best of best is better and much better than the average of the community. Okay, which is good news, I think, if you are working in computation these days. Of course, there's a big difference between before and after AlphaFold, and CASP and Capri have been highlighting that. And these experiments are very important in defining what is the state of the art. So if you are working in methods, new methods to model interactions or model structure, you want to put your method to the test in such experiments. But you can see that there are challenges. large domain motions, antibodies remain challenging. AlphaFold 3 is doing much better on those, but they reach about 50% success rate, but at the cost of a massive sampling again that not everyone can afford. And scoring, as I just showed you, is a big issue. Big thanks for this part to the Capri Management Committee and all the participants and all the people who are providing targets, or advisory board, and then the CAS committee, which has been run in the early day by John Moult. Okay, so this was just an overview of where is the field standing when it comes to structure prediction of complexes. Now we're going to move to what we have been doing in terms of integrative modeling of complexes with Haddock. So Haddock is an integrative modeling platform where what distinguishes it originally when it came out, it was published in 2003, so we have been developing Haddock for almost 25 years by now, is that we are using as much as possible experimental information to drive the modeling process. So we are not doing ab initio modeling, no, we want to use information when available to drive the modeling process, to bias basically the search. It started from NMR data, but there are many different types of data that you can use. And it also has modes where you can run in ab initio mode without data, but it's not what it was designed for originally. Over the years, we started with proteins, and over the years, we've been adding support for many different types of molecules. So you see protein peptides. So peptides are simply small proteins, but they have more flexibility or a flexibility challenge, conformational change challenge, so we treat them slightly differently. Nucleic acids, both DNA, RNA, glycans, glycosylated protein. This is something that we added support during COVID. There was because glycans are very important in shielding the spike protein from our immune system. Small molecule, you can do small molecule docking with Haddock, and this was there from the early days because we needed to have a way of describing cofactors, for example, that might be very important also for the binding in some cases, if they are the interface. More recently, we added shapes, so an arbitrary shape, and we added also more coarse graining to be able to move to larger systems. So you see here such a shape, two planes with fake particles. What could that represent? What kind of biological system could that be? Sorry? A membrane, yes. So that would be a very simple and coarse description of a membrane. And say you are modeling two membrane protein associating, you might say, well, they should be in between those planes, or they should overlap with the beads. The beads could be the phosphate atom of your lipid bilayers. Or if you are modeling a membrane protein binding to a soluble protein, you could say one should be here and the other one should be either on top or at the bottom. So we can use this shape to represent arbitrary object. You could also think of this shape here as being a reconstruction from a Sachs experiment, where you will get a shape of a molecule, a shape of a complex, and then you say, when you're doing your docking, your molecule should overlap with this. This will not have any energetics. So this is not generating classical energies, but it can generate restraints to position your molecule in space. And we use that also for small molecule docking. So we have a very efficient protocol for small molecule docking, which use basically homologous systems to define shapes, and then you are docking your molecule into those shapes inside the protein. In terms of restraints, we have a lot of restraints that are NMR related. If you remember my introduction showing you where I'm based, we are in a big NMR group. So from the early days, we've been implementing a lot of NMR data into Haddock, but we also have support for cryo-electron microscopy, more if you are at a medium resolution. If you have high resolution, you don't need to do this kind of modeling. Symmetry is something that we can use as well to basically restrain the search. So how do we do the docking classically in Haddock? So Haddock has 3 stages, and it uses a combination of energy minimization and molecular dynamic simulations. Okay, so it's a physics-based search. So in the first phase, we're going to treat the molecule as rigid, rock solid, and we're going to minimize the system based on the information that we have. This is where the docking takes place. This we can do 10,000, 100,000 times typically. And then we're going to select a fraction of those models. And then we're going to apply a simulated annealing where we introduce flexibility in the system to refine the interface of those molecules. So this is a protocol which is also used to refine NMR structure when you do NMR structure calculation or when you do crystal structure calculation. So we start at high temperature and we cool down the system in multiple steps, and we do molecular dynamic in torsion angle space at that stage. And we use to put then the molecule in water, a layer of water, so it's not periodic boundary condition like if you do MD and run a very short simulation. These days we just minimize the system. So this is how it looks like. So here you see the energy minimization. So this is fast, this is seconds or less. So the degrees of freedom here are only rotation translations of the molecule. The molecule are purely rigid. You might start your docking from from an ensemble of conformations. So if you pre-sample conformations by MD, by elastic networks, by taking different conformations from your crystal structure or multiple AlphaFold models, things like that. Then you see this torsion angle dynamics. The advantage of working in torsion angle space here is that you can freeze easily degrees of freedom. So the degrees of freedom here are rotations around bonds. So you can say that your molecule is rigid except at the interface where you allow rotation along the side chain and then you allow rotation along the backbone as well. It means that the molecule can still move freely in space, but you have part of the molecule which is frozen. When you do classical molecular dynamics, when you start your equilibration process, usually you apply position restraints on your system. But it means if you put position restraints, the molecule cannot move in space anymore. Okay, so here we have this torsion angle space is useful for that. And you see that small conformational changes can be now modeled here. You see this helix here, or the loop. You see the side chain moving now, and then in the second phase, we have a backbone moving, and this loop is going to flip over. So this is a small binding-induced conformational change. And in the final part, we used to put a layer of water on those and run extremely short molecular dynamics. So you're speaking of 10, 20 picoseconds, so it's peanuts, but it was optimizing a bit the energetics of the system. And now again, it's still there. You can turn that on if you want, but we only do minimization. So we have a classical force field when we do all those computations. It's based on the OPLS force field. And this is the scoring function. So the scoring function is how we rank the model. When we calculate the model, we have a full energy function. When we rank the model, we use a simplified energy function, which has different terms, and they have slightly different weights at different stages. So this will be the function at the end of the protocol. And what do you have in there? So you have intermolecular energies, so electrostatic and van der Waals energies, the same terms that you have in molecular dynamics, but only the intermolecular component of those. The electrostatic is down-weighted. We have an empirical term, which is the dissolution energy. So this is the price or the bonus that you get by removing water from the interface. Okay, if you have an hydrophobic surface, two proteins binding to each other, you have to remove water during the binding process. If you remove water from an hydrophobic surface, you gain energy usually because the water is going to go into the bulk of water, and it's much happier in a bulk than on this hydrophobic surface. Water and oil don't mix, okay? So it's not an energetically favorable interface. So by removing water from hydrophobic surface, you typically gain energy. And this is what it's measuring here. If you remove water from charged surfaces, usually you have to pay a price, okay? Because then, so this is a combination of basically there will be positive and negative contribution. So this can be positive or negative. And the last term is basically representing the data that we put into the modeling. So this will be the experimental information or the bioinformatic predictions that you put in it. This simple function has survived now 25 years of optimization. So it was optimized at the time 25 years ago on 5 complexes doing a grid search in parameter space. Since then we had a lot of data and so today you say this is machine learning, okay? I'm doing machine learning here. That's a very simple linear model. And students want to do machine learning these days, so we give them a lot of data when they come to us and say, okay, go beat the scoring function of Haddock. and they come back very proud and they say I have a much better scoring function. Instead of 1.0, we should use 0.97732. This scoring function is not perfect, but it's robust. So that's why we still have these very round numbers. The only thing that we change from time to time in this part is that we increase the van der Waals. When you're doing small molecule docking or glycans, we increase the weight of the van der Waals here. But otherwise, it's still pretty much what we're using. And the other advantage of having a simple function is that we can score different types of interfaces at the same time. So you're not only doing binary complexes, you might be building a protein-protein nucleic acid interface, meaning that in your complex, you will have protein-protein interface and protein nucleic acid one. And if you start having scoring functions that are fine-tuned for each different type of interfaces, the scoring becomes complicated. So in principle, the physics is the same. There is no perfect scoring function out there, otherwise we would not have the problem that I've shown you in a CASP, okay? We're also doing deep learning. You heard about AI and machine learning yesterday. You heard about graph neural networks, 3D convolutional neural networks. So we started doing 3D convolutional neural network where we are representing the interface of a complex as a 3D image. And then we train a network to distinguish what will be a good from a bad solution. And then we switch to using graphs because graphs are rotationally equivariant, so it's easier for the training. And then you associate, so the edges, so these are the connection, will be the connection, the amino acids that are close in space in your interface. And we distinguish between intermolecular connections and intramolecular connection. And the feature, the nodes are basically the amino acid in your system. And you associate features to those, which could be the type of amino acid, polarity, charge, but also this position-specific scoring matrix, which is basically a measure of conservation of that particular amino acid. For this, to calculate this, you need multiple sequence alignment. So more recently, we have been using large language models. So ESM2 from Meta, you don't need a multiple sequence alignment anymore. So from a single sequence, you can get these embeddings. So it's much faster for scoring purposes, and it performs as well as the multiple sequence alignment. So that's the latest version of DeepRank. Now, most of our users are using Haddock for its web server. We are not going to do that today in the tutorial. We're going to use more a new version, which is not available as a web server, but this is the entry point. So this is used quite a lot. It has been operating since 2008. And we can do that because we have access to these high throughput compute resources. So when you use Fugaku, we are speaking about HPC, high performance computing. Here we are speaking about high throughput computing, and the difference is that we are sending a lot of small jobs to many computers distributed around the world. And each computation is very short, but we have to do a lot of them. Again, when I told you we do docking, the initial phase, we sample 10 or 100,000, and each of those computations does not depend on any other. So we can distribute those. So the server sends in the order of 20 million jobs per year to the HTC resources of the European grid. This is our current user map from last week. So 76,000 users from more than 182 countries. Do you know how many countries there are in the world according to the United Nations? Sorry? 193 or 196 or something like that, okay? So we are missing about 10 countries. And probably Vatican is one of those, and San Marino is one of those, I don't know, but okay. And we still have to work a bit on Africa, but otherwise we're doing, and a little bit here. So these guys are hard to get, but yeah. So, yeah, most of our users these days, so India used to be the winning country, but I think now that China has taken over slightly, but these two are representing a major fraction of our users. This is the cumulative number of users over the years since we started in 2008. So AlphaFold occurred in 22. So I would have expected to see a slowdown in registration after the outcome of AlphaFold because, you know, this has changed the way we do structural biology. We don't see that. So it's still exponential in growth, so which is good news for us. But it also means maybe that people are also taking AlphaFold model and refining them. So you can also use Haddock to refine models, and you don't have to do docking from scratch. But if you want to refine the models, maybe remove some clashes and get some energetics, you can do that. This is the number of monthly registration, how many users are registering every month for the server access. What you don't see here, so this is now starting January 24. What we also observed, if you go to the period where during COVID, so during COVID we got a doubling, almost tripling of registration. So we went into lockdown, we could not do experimental work anymore, and people realized that you can do computations. So the number doubled to triple during COVID and remain at that level. And at the beginning of last year, again, we see almost a doubling in usage, and I don't know where this is coming from. Okay, we've been working quite a bit on antibodies in the last couple of years, so maybe that's the result of publication that we had, but it's difficult to tell. So it is again a significant increase. This is the number of docking runs that are processed by the server every month, so that's on average around 15,000 per month since last year. And this is the distribution of users. So you see out of 76,000, 40,000 are coming from Asia. Okay, so this school is representing a large fraction of the usage. So this was classical Haddock. And now since several years, in the context of a European project called BioExcel, Center of Excellence for Computational Biomolecular Research, we have been working on a new version of Haddock, and that's the one that you're going to use in the tutorial later on. Before I go on, are there any questions at this time? Yes.
Thank you so much for your talk. Maybe I would like to follow up about the scoring function, like about the refinement of PPI that you mentioned. So say that we refine the complex docking from the AlphaFold, like with the PPI docking such as a Haddock. Would the scoring actually still be reliable after the refinement, and perhaps would that be closer to the ground truth?
So well, it's going to give you some energetics. It might remove some problem at the interface. AlphaFold 2 still has some clashes often when you look at the interface. AlphaFold 3 is much better in that sense. So AlphaFold 3 has learned a bit more physics, I think, and chemistry. There's no guarantee that the Haddock scoring will be perfect. We're also participating in CAS-CAPRI as scorers only, and we are using our standard Haddock free pipeline to score models, and we didn't pick out the right models. So the scoring, no, it's not solved yet, but it might give you confidence, and it might also give you interesting information about the interface, but it's not a solved problem. Okay, so Haddock scoring is not the answer to all problems. Okay. More questions?